Primitive cell symbiosis possesses a genotype duplicate and a duplicated energy preparation system. We are biological hermaphrodites in the evolutionary sense of life. All bioenergy and biochemical processes, above all naturally those in the mitochondria too, depend on a varyingly intensive negative redox potential as a biophysical prerequisite for complex proton and electron flow. This is guaranteed chiefly by glutathione, a tripeptide unique to quantum physics that avails its central molecule, the amino acid cysteine, via the hydrogen sulfide group, especially freely convertible protons for all detoxification services. Raum plus Zeit. What are the consequences of these findings in understanding the causation of cancer, the causes of AIDS, and therapy for cancer and AIDS? Kremer. The consequences are fundamental. In the case of cancer and HIV positive, that means increased production of many specific antibodies. In full-blown AIDS, that is, intracellular fungus, protozoa, and mycobacteria infections as well as a few really existing virus infections, it means ulcerative colitis, severe traumas, burns and other systemic and chronic diseases. We have a systemic lack of cysteine and glutathione as the result of excessive cysteine and glutathione use, as with the nitrile compounds above, and/or lack of cysteine uptake and/or disturbance of new cysteine synthesis from methionine in the liver, for example, through folic acid inhibitors such as Ciptram, and/or disturbance of new glutathione synthesis, toxic and pharmatoxic due to a variety of substances. The organism suffers from a striking lack of freely convertible protons. Under current civilization conditions, the organism must cope with more than 60,000 poisons in the glutathione system. Transformation to cancer cells can develop through a shortage of glutation when the mitochondrial respiration chain's reserve capacity for ATP production is reduced insidiously below a critical level of reserve energy, apparent lack of oxygen, pseudohypoxia. The primordial genome portions in the nucleus genotype function in this case as a proton shortage memory. In genetic and supergenetic terms, it develops into highly complex counter-regulation. Alternating switching with the mitochondria is blocked. The cells can no longer switch back after cell division and remain caught in the division cycle, nor are cancer cells transformed in this way any longer able to die a programmed cellular death, because the mitochondrial gates that would need to open remain closed due to intense counter-regulated no-gas synthesis. Crucial here too is the circulatory calcium exchange formed between the mitochondria and the cell plasma that is also handicapped. Cancer cells have a striking embryonic character in many respects, thus it involves a surviving re-switching mechanism on the disorganized gene and energy program, a regression that could not be explained in the past by malignant coincidental mutation. From an evolutionary medicine standpoint, concerning the cell symbiosis processes, one can comprehend cancer cell transformation if one understands the laws of coevolution. Raum plus Zeit. Can the blockage of defective alternating switches for cancer cells be reversed? Kremer. That is the cardinal question for therapy. The disappearance of Kaposi sarcoma after eliminating azathioprine, that caused high use of glutathione as well as all nitrile compounds, suggests this. Yet in the meantime we have an abundance of other evidence. It was also possible, in animal experiments, to prompt tumor cells as well as metastases to disappear completely by stimulating synthesis of no gas. Undoubtedly, the most impressive is the success in healing cancer by balanced high dosages of cysteine and glutathione to regulate the potential for redox by means of preparations with good bioavailability. Raum plus Zeit. Does glutathione therapy suffice, or must other measures be combined with it? Kremer. Cell symbiosis therapy to harmonize redox with equal amounts of cysteine and glutathione is a must as basic therapy. Yet cancer is a Highly individualized and highly complex event. Countless studies during the last 10 years have proved the effectiveness of various therapeutic options in counter-regulating cancerous cells by non-aggressive inhibition. The art of healing through counter-regulated cancer cells calls for carefully thought-out interplay between gas pedal and brake, so to speak, since basic understanding of cell symbiosis programmed by evolutionary biology was not yet sufficiently advanced. Past cancer therapy lacked broad-based testing of a systematically combined and rationally assured overall concept of biological compensation therapy, or expressed in traditional terms, harmonizing of the yin and yang. In the meantime, however, we did understand why cancer patients died mainly from cachexia's tuberculosis syndrome as the result of a nitrogen and energy imbalance. If you ask cancer specialists how to stop their cancer patients' cachexia, even today you will hear, by supplying high-calorie protein. A study in German clinics found half of the cancer patients to be undernourished, as one can verify in the standard works of AIDS medical officialdom. Therapists treating AIDS as well as cancer have for decades confused cachexia, called HIV-related wasting syndrome, in the case of AIDS patients with a chronic state of hunger. They have not understood why the protein was mainly excreted as urea. On one hand, cachexia results from a proton deficit due to lack of cystine in the liver that leads simultaneously to a lack of glutamine and arginine as well as to an increase of glutamate in the plasma. On the other hand, recycling in the liver produces the glycolysis product lactate. This occurs due to a 20 times higher glycolysis decomposition by fermentation in the cancer cells, an excessive use of protons, and higher investment in energy than was obtained originally as energy from fermentation of glucose. These feedback processes are regulated via type 2 cytokines, communication protein that are synthesized by force due to the glutation shortage and prevent in the net result protons from splitting out of the cystine. Thus the primordial anaerobic principle of low fluid proton fixation also shows up with cachexia in comparison to the high fluid proton floating of intact cell symbiosis. Check the laboratory findings notes of clinics and medical practices. Then it will be clear to you why the causes of systemic amino acids dysregulation are usually misunderstood and inadequately balanced. Raum plus Zeit. Can biological compensation therapy dispense with chemotherapy? Kremer. In principle, yes. Chemotherapy seeks above all to inactivate the cell division process, yet it affects the mitochondrial structures. Primarily, as descendants of eukaryotic bacteria, they possess no protective proteins and no effective repair mechanisms for their genes. However, they are many times more sensitive to peroxide chemotherapy as, for example, genes in the nucleus that are especially protected. During the long course of evolution, the mitochondria have functioned very well among animals living wild. DNA defects in mitochondria have been detected rarely, while the list of congenital and acquired mitochondrial illnesses among human beings from Alzheimer's to Parkinsonism and severe heart myopathy becomes ever longer. The problem of any chemotherapy is that cells in any tumor are found with variously intensive degrees of counter-regulation. Thus one can use chemotherapy to kill part of the cancer cells. That's called remission. Other cancer cells must encounter intensified counter-regulation. This is due precisely to the intentional simultaneous attack on the mitochondria. It also applies to cells that are not yet transformed and still exist in the compensated state of cell dyssymbiosis. As a result, metastatic cells are secondary. Tumors can be selected. Cancer patients who have been subjected to biological compensation therapy before and during chemotherapy report less side